Okay,
Okay, perfect.
so it's ready. Um, yeah, let's start. So the main purpose of this tutorial or this talk is to provide you an overview of the different strategies that are available, or some of the strategies that are available in enzyme design for predicting active site or distal mutations. And of course, the main purpose is to show you how the DINACOM works. But before that, I think it's good that we do some, like, let's say, that we see an overview of the different strategies and we remind some key concepts. So if we go to the next slide, I guess now you are looking at the protein fitness landscape slide. Is that right? Um, I guess it is. Okay. Yeah, as you already know, when you want to design an enzyme, we always do the analogy to climbing a mountain. So you start at the valley, and you wanna go all the way to the top to maximize the property that you are interested. And to do so, there you can use different paths. So you can click on the different paths. And basically, all strategies for designing enzymes are composed by these three key points. First, selection of mutation points. So how these mutations are, or these positions that are going to be mutated, will be selected. In here, we have how the different methods differ. Then we have generation of variants. Yeah, of course, this can be done computationally, but also experimentally. And then you have the screening for activity, which is also quite key for, especially when you generate a massive amount of variants. Okay, the next slide is the usual slide that you have seen multiple times. So the main idea is, okay, how can I move from an enzyme that is inactive and I make it active? For the reaction that I'm interested, maybe there is no natural enzyme that catalyzes the reaction that we are interested, or maybe there is one, but the substrate scope of this enzyme is not as broad to accept the substrate that is interesting for industry. So yeah, the main idea is we want to turn this enzyme that is inactive into a highly active one. And how can I do that? Well, you know that there are experimental strategies and there are computational strategies. And these different strategies can be classified in these different non-rational, semi-rational, or rational, depending on how much knowledge is required or is for performing this experiment. If you don't hear me or if there is something wrong with the internet, please let me know. Okay.
For now, it's perfect, I think.
Yeah. Okay. Fine. So yeah, I guess you are looking at the correct slide also. So we can move. I will quickly summarize these main strategies very quickly because I want to go directly to the details of these different computational strategies. So if we move to the directed evolution slide, so you know already, the Nobel Prize in Chemistry 2018, and this technique mostly revolutionized the biocatalysis field because thanks to that, variants that have very high activities can be generated. So this strategy is based on this random mutagenesis. So you make the polymerase to make errors. And with that, you get a huge library of variants. And these variants have the advantage that the mutations are distributed all over the gene. So it's not restricted to active site, but it can incorporate mutations everywhere. And of course, this is it's a kind of expensive technique. It requires the generation of multiple variants and also the screening of multiple variants. So it was nice that in these 2 weeks ago, Mikel already talked about these different screening methods that are used in this strategy. And one of the points or the bad points about directed evolution is that usually or the process it's better if you have some initial activity. So you should have an enzyme that has some initial, although it doesn't matter if it's very minor, but it should have some initial activity. And for that reason, this strategy is best if it's combined with some rational strategies at the beginning. So most of the enzymes that have been dramatically evolved through directed evolution have had some rational approach involved, let's say, um, and and yeah. Um, and with that, then you can, with this very small initial activity, then you can boost it and have, variants that are very, very active. So that leads me to the next slide, which is this kind of, grids, strategies with the casting and the ISM, the iterative saturation mutagenesis. Well, in these strategies, of course, you need to have, so in direct evolution, in principle, if we don't take into account that some initial activity should be present, in principle, you don't need to have any kind of knowledge about the enzyme. You don't need the structure, for example. But in this kind of semi-rational, rational strategies, of course, you need to have information about the enzyme. And usually you take the X-ray structure. So you have the X-ray maybe with something bound or maybe with nothing bound. It doesn't matter. But you took the X-ray, you know where the active site is. And with that, you manually select which positions you want to mutate. So you say, okay, so taking into account your chemical intuition and also depending on the problem that you are interested in solving, maybe you want to make space or maybe you want to that the enzyme accepts the other enantiomer. So depending on the problem, then you will manually select which positions in this active site will be subjected to mutagenesis. Okay, and this is the idea of this semi-rational. And then you can try like all possible amino acids or just a selected set of amino acids in these positions. Okay, so the key point here is that, okay, with the structure, one can define which positions can be subjected to mutagenesis and then generate these variants experimentally, or you can also do it computationally. And this leads to the rational, so the next slide. So the same strategy can be followed using a computational program. In this case, there are different options. So Rosetta is the most famous one, I would say, by David Baker. But there are also others. Orbit, for example, that was developed by Stephen Mayo, now called Triad. So there are these programs, computational programs, that have like the same idea as before. If you want to do a redesign, you will check the X-ray structure, manually select which positions you want to mutate, or you want Rosetta or Orbit to mutate, and you will use the program to make these mutations and to select according to the score which is the one or the variant that has the lowest in energy score, for example. So that's the regular strategy for these computational approaches. So here I said, well, and of course, in the particular case of the Inside Out protocol that was developed by Ken Haug and by David Baker, so with this protocol, if you focus on the whole thing that is here shown, so it starts with a thiozyme. So in this case, so you do the whole cycle if you don't have an enzyme that has some initial activity. So imagine that, well, or they wanted to do a chem eliminase or a Diels-Alderase. So there was no natural enzyme to start with. So they used this approach, or they developed this approach to try to put to design the active site. So they did the thiozyme, that is as you know, short for theoretical enzyme, where you use DFT, and with this DFT, you start to imagine how you would like to stabilize the transition state. So you position the different side chains of the amino acids, and you optimize, and you get the perfect arrangement for stabilizing that transition state. And then this arrangement is put, let's say, in a protein scaffold by Rosetta match, and then you have, in principle, let's say, at this point, you can start the redesign. So trying to incorporate more mutations in the active site pocket to make that this transition state is better stabilized. So that's the inside-out protocol. And the original idea of the inside-out protocol. Usually most of the attempts to design new enzymes are mostly redesigns, in the sense that you start with an enzyme that already has that has already the catalytic residues that are of interest for you, and you introduce some mutations to it to make it more active for the other substrate or for whatever. And what I want to highlight here is that in these protocols, it doesn't matter if you use Rosetta or if you use Triad, you always, so they are focused on the active site. So you know where the active site is, and then you select, or you manually, although I know that there is an automatic way of running Rosetta. So you can let Rosetta automatically decide which positions to mutate. But experience has shown us that this is not the best option, as Rosetta will mutate everything it tried. So the best is that you constrain the program. You define some rest file that is called, where you say, okay, mutate please this position, that position, that position, that position. Also, experience says that it's best if you say mutate this position to this set of amino acids, like polar amino acids or non-polar, or so you should impose to the program some knowledge that is required. And as I said, always mostly active site. And you do that, and this strategy is not that different from the read strategy, in the sense that usually you take the X-ray structure, you check where the active site is, and then you manually select which positions you are going to or you want to mutate. And the original designs or the original idea was always on the static X-ray structure. Although at the end of the process, as it is said in this slide, some MD simulations were done for ranking the variants and for deciding which among the multiple variants that you get will be the ones that will be tested in the lab for activity. From this original approach, of course, some improvements and some alternatives have been proposed. For example, to improve the point that is an static design, so to make it a little bit more dynamic, the Rosetta backgraph by Tania Corteme was introduced, and in this backgraph move, basically the phi and the psi angles of the backbone on the designable part of the enzyme will be a little bit distorted to have a little bit of flexibility. So that is one option that is already implemented in Rosetta, and that if you have Rosetta, then you can use the backgraph moves. So you can tell Rosetta to do that. There are also other strategies that have been followed. And it's like, okay, instead of using the X-ray structure, why don't we use an ensemble of structures? And this ensemble of structures can be generated via, for example, MD simulations, via normal mode analysis, or again from modifying the psi and psi angles. And here is when there are different approaches that I would like to show you in this tutorial. Well, the idea, if you click, Fred, and now you should be looking at why mutations only focus on active site, why not accounting. So if you are looking at this slide, um, so the so these are the key questions that we are asking ourselves. So it's like, okay, why should we only focus on active site mutations? Why can we, I mean, if we want to do a mimic of directed evolution, we should be able to predict also distal. And why we are interested in distal? Well, because, as I'll show you later, distal mutations are key for improving KCATs. And the other question that we are trying to solve or to answer is why the conformational dynamics, the conformational heterogeneity that enzymes present, why this heterogeneity cannot be used for designing. So can we identify the mutation hotspots based on the conformational dynamics of the enzyme? First question. And second question, can we, depending on the conformational dynamics and which conformational states are or the enzyme presents already, so can we use this information to select the best scaffold for redesigning? And this is, for example, related to this ancestral reconstruction by Kamerlin that they are recently doing. Okay, so these are the questions that I want to, that we want to answer. And as I said, I would like now to show you different strategies that are quite recent strategies that are being applied in this computational enzyme design field that try to use this multi-state design, also in the sense that they try to incorporate more than one structure in the design process. And also I want to highlight and check whether these new approaches are focused on active site or far away from the active site, and how these different mutations are selected. So if we move to the next slide, now you should be looking at the computational design of multi-substrate enzyme specificity. So this is a paper by our colleague Roberto Chica. In this case, so you know that Roberto Chica has developed is applying this multi-state enzyme design. So usually they are using triad, as Miguel Angel has explained us in several occasions. So they are using triad, and the design process is not based only on a single structure, but rather on an ensemble of structures. And this ensemble of structures are taken from short MD simulations. So that's what they are doing. In this paper last year, they also show that it's that apart from multi-design, you can also do multi-chemical state analysis design, let's say. And in this case, I like this study because they try to redesign an amino acid aminotransferase, and this aminotransferase is a PLP-dependent enzyme that has quite a complicated mechanism. So, so the reaction goes through several intermediates. So you have the internal albumin, the external albumin, the ketimine, etc. So multiple chemical states that in principle, when you want to design the enzyme and redesign it, you should be taken into account. So the original idea of the in set out, like, okay, you have one transition state and you try to incorporate that transition state is very, it's nice, but the reality is that usually there are many steps, and in principle, all steps should be, should operate, should be working. So you want your enzyme to do, to catalyze all the steps. And of course, you can always focus on the rate determining step, but maybe by introducing mutations, you change that. So I think it's a nice strategy to try to incorporate multiple intermediates in this design process, apart from the multiple structures. And well, here I highlighted some key points about the strategy. Well, one thing is that although they use multiple structures, as I said, and also they use multiple intermediates for the design process. They well, when they have to introduce the mutations, if you check the blue box, so basically the strategy that they use is again not that different from the what I was explaining. So they look at the active site, they take the PDB, and then identify which active site residues might be involved in substrate binding. And they select those positions for mutagenesis. So if you go to the next slide, basically once they have selected which positions, so you see on the right side, for example, tyrosine 31, valine 109, etc. So these are the positions that were subjected to mutagenesis. And on the left side, you have the protocol that they use. So in this case, they design, or let's say they design a new protocol that tries to maximize or tries to identify the best sequence, not for the best sequence for all chemical states that are involved in the process. And if you look at the right side again at this position probability matrix, you see that, for example, at the PLP bound state, you see that there's a, I don't know, for example, at position 31, there's a propensity, a high propensity of putting a phenylalanine or a tyrosine. And however, if you look at the histidine, when histidine is bound in the active site, then you see that also there is a methionine, and this methionine also appears in the glue chemical state. So you see that they did this kind of analysis, which are the best amino acids at each chemical state, but at the end they try to group all these different possibilities in the his glue PLP, and they identify which are the best amino acids at each position that in principle should favor all chemical steps. For example, at position 126, it's pretty obvious that a tryptophan should be included. Yeah. And by doing so, they were able to increase the enzyme, the enzyme activity towards a non-natural substrate by 200-fold. And thanks to the development of this computational approach, experimentally, only 32 mutants had to be screened for activity. And they say that, of course, this is very beneficial because you don't need to do many sequence alignments, you don't need to do costly MD simulations. Simply by applying this protocol, you can improve the enzyme activity towards this non-natural substrate in a highly complicated mechanism. And so, summing up, in this Chica's approach, you have that, okay, there is a multi-state design because an ensemble of conformations is used. Usually, these ensemble of conformations are not that different because they come from short MD simulations. They also allow this multi-chemical state design because different intermediates are taken into account. However, in this approach, we have mutations at the active site, and the mutation selection again is manual based on our experience or our chemical intuition. Okay. I guess everything is working fine. So I will go ahead and go to the next one. The next study that I wanted to highlight here is this KDE, so computer-aided directed evolution of enzymes. So this is a paper by Cameron Lin, where they develop a strategy to mimic directed evolution. So they propose a protocol which they validated with an enzyme, triose phosphate isomerase, an enzyme for which they have a lot of information, there's a lot of mutagenesis. There are a lot of mutagenesis studies, so they have a lot of information to use for validating this new approach. And this new approach is based on this empirical valence bond, so this semi-empirical COMMM method. And as you know, that this was developed by Warshall. And here you have the figure 2, the main idea of Warshall's reorganization energy. So basically, well, you have the difference of water or an enzyme in catalyzing the reaction. So you define two different states, one for the reactant, the other for the products. And you see that the difference between water and the enzyme is that the barrier is smaller, basically because the reorganization energy in the enzyme is smaller than in water. So water, the dipoles need to be reoriented to stabilize the transitional state. In the enzyme, or the enzyme active site, NH backbones are already properly positioned for doing that, so less energy is required. And with this, and here below you have figure 3, where they show the delta G, so the barriers of different different enzymes. So here you have, for each column is a different enzyme for which they have the experimental value for the barrier, and how EBB performs in reproducing the experimental data. So it's a powerful approach that it's not that expensive, because here they propose to apply it massively to generate multiple variants, and it provides a good estimate of the experimental barriers. So they propose this protocol shown in this figure in the left, where they do some initial mutagenesis alanine scan, then they do this user-directed mutagenesis. So they say, okay, I want to mutate those positions, and in those positions, all possible amino acids are tried, and with EBB, you compute the barrier, and you see how this barrier is affected, whether it has been reduced or not. And you do that. You do this several times until you get, let's say, a variant that has a smaller activation barrier. And if you move to the next slide, so here you see, for example, some results where some positions were subjected to mutagenesis, and they computed the activation barriers, and they use, as they did MD, to have several snapshots, so they have different barriers for each snapshot. So that's why there are these bars, these deviation bars. And so with this strategy, it's multi-state, because you can you have an ensemble of conformations similarly to Chica, let's say. So you do really short MD. I think they did 8 nanoseconds MD, and from these 8 nanoseconds they took different snapshots, and different snapshots they compute the barrier with this EBB. The mutations are also located on the active site, and in this case, the mutation selection is manual, and the main focus is on reducing the activation barrier. Similar to that, they did this KDE in this triose phosphate isomerase, as I was saying, and basically this was proposed, or this was shown. Well, it was demonstrated that they could validate the experimental observations. So they were proposing that this kind of approach could be used to design enzymes. Um, to design enzymes. Similarly, in this line, of course, we have Warshall's approach. So he's the main, the EBV developer. So there is this paper in ACS Catalysis, very recent, just accepted paper, where also they proposed an approach for computer-aided enzyme design. And this approach, again, is based, well, they say that it's a combinatorial approach for exploring conformational space and activation barriers. Why they say conformational space? Well, because they consider different side chains, different rotamers for the side chains. Okay, so and that's why they say that they are exploring the conformational space. And the schematic you have in figure 4, the schematic presentation of the stages and the routines that this protocol is using. So they use the protein structure, PDB, so they relax the structure for doing the EBV protocol. They extract the active site, well, for calibrating the parameters. Then they do these EBV calculations in the protein. They relax the structure. They can, they generate different structures, and they also take into account different rotamers for the different amino acids, and then they calculate the delta G's, and they do this multiple rounds. So it's a similar strategy, I would say, to Kammerlin's approach. And in this case, they validated this approach using chemoeliminases, the AG3 chemoeliminases, and also haloalkane dehalogenases. So again, in this case, if you click, now you should be looking at the teal box. So in this case, well, multi-state design, well, it's similar to Rosetta. So in the sense that only side chain, different rotamers for side chains are considered. The mutations, again, restricted to active site, and which positions to mutate is also manual. So you, with your chemical intuition, you select which positions you are going to mutate. And again, the focus is on reducing the activation barrier. If we move to the next slide, I also wanted to highlight here the Pele-based approaches. So you know that Pele is this Monte Carlo based program developed by Victor Guayar at BSc. And they also proposed that Pele could be used for doing this in silico guided directed evolution. Well, in this, you have in this figure the protocol that they propose. In this case, as they propose to identify try to mutate all residues within 10 angstroms of the substrate. And as they perform these Monte Carlo simulations with the substrate not in the active site, then as the substrate gets into the active site, then also mutations can be introduced in the tunnel, let's say in the substrate tunnel. And he has some papers on showing that. But usually here, I think they put the substrate bound in the active site and basically identify everything that is within 10 angstroms of this substrate, and they try to mutate all positions to all possible amino acids. They do the mutation, they relax the system with Pele, and they use Pele's interaction energies to rank the different variants. And while this strategy has been, for example, now they are using this strategy, well, they have used it for designing multiple metallo-based enzymes, but very recently there is this paper in Nature Catalysis that you have seen where they do these plurizymes, I think they call it, where they evolve one enzyme and introduce two active sites, let's say in the same enzyme. Well, in this kind of idea, in this approach, while multi-state, yes, could be considered multi-state because there is an ensemble of conformation taken from these Monte Carlo simulations. The mutations are in the active site. Also, in some studies, they have also positions located at the entrance tunnel, for example. Um, and in this case, it's like, okay, everything that is within 10 angstroms, so it's like brute force, so automatic, the mutation selection. And the main target is to have better Pele interaction energies. And well, in this case, I was saying, okay, maybe following this, you could also have substrate inhibition if you have a substrate that bounds very strongly to the active site. But the truth is that it works, so they have used this Pele for designing different enzymes. Of course, tunnel, no? If I say tunnel, then everyone has in mind, next slide, sorry, Kaver-based approaches. Here I put a paper that is not that recent, it's quite old, but I think it's nice. So it's based on these haloalkane dehalogenases that present different active sites, more open, more close, and they try to engineer, so they try, so here in the figure, for example, they say, okay, if you put the 11 amino acid of 11 amino acid and 8-point mutation from a highly enantioselective DBJA into the weakly and enantioselective DHA, it will yield weakly enantioselective haloalkane dehalogenase. However, they show in this paper that by introducing 5 mutations located in the tunnel area, let's say, then you can you can have a strongly enantioselective new haloalkane dehalogenase. In the Domborski lab, they do multiple strategies. Well, in this particular case, they use multi-state design. Well, not really, because they based on the X-ray and this tunnel analysis with Kaver. Although, as I said, they are also doing different. They are following different strategies. The mutations are in the active site or in the tunnel, and also they can, as mutation selection, they can use Kaver information, like for example, residue in the bottleneck, to select which positions to mutate. And also in this particular case, they evaluate the different variants by looking at these near attack conformations in short MDs. But of course, this depends a little bit on their study. The next, of course, is by Janssen. Next slide. So you know that, Janssen is a developer of the Casco, Fresco semi-rational approaches. In this part, and here I put this recent, quite recent example. Well, they redesigned some enzymes for enantioselective hydroamination. And here in this figure 1 of the paper, they put the protocol that they use. So as you see, they use the crystal structure as a starting point, and they use Rosetta design. So again, it's very similar to the Rosetta design features that I was talking about a few slides ago. Is it multi-state design? Well, it's based on X-ray and in Rosetta, so it's not really multi-state, although they do MD to refine and to check these near attack conformations to see whether they analyze all these distances that are shown in figure 1C and see whether these are, the mutations have improved these distances along these short MDs in multiple replicas. The mutations are in the active site, and the mutation selection is again manual. Okay, so if you now move to the next slide, so at this point, we have so far seen different approaches that have been developed in the recent years that try to solve, let's say, the enzyme design problem following different strategies. However, I think the main idea or the main message that I wanted to provide here is that most of these strategies, are based on introducing mutations in the active site of the enzyme, and mostly the positions that you are trying to mutate or that you try to mutate are manually selected. One of the questions that I was saying that we want to answer is why should we only focus on active site mutations, and why not trying to introduce distal mutations? And the question is, okay, why should we care about distal mutations? Well, I really like this paper by Professor Kell, where they highlight the importance of distal mutations. So it's a review paper. And here in the first paragraph, the distribution of KCAT values among natural proteins, so they say that, okay, although we always say that enzymes are the best catalysts, the truth is that they are not all of them are working that efficiently. So they don't have these massive KCAT KM values, all of them. Few of them have, but not all of them. And basically, one assumes that this is because evolution simply had no need to go to higher rates, let's say. Here also he highlights that usually in industry, well, these enzymes that are used in industry, you have so you use high concentrations of the substrate, and so Km. So he says Km is largely irrelevant because the concentrations in use are huge, and so the focus is on KCAT. And here also he says that while directed evolution has been shown to be capable of improving enzyme turnover numbers significantly, examples show that even the poster child examples like sitagliptin or halohyrin or dehalogenase LOVD, etc., have turnover numbers that are very poor compared to those of typical highly efficient enzymes. And also he highlights that while in the other paragraph, the importance of non-active site mutations in increasing KCAT values, say, okay, so our focus is on KCAT, so how should we improve KCAT? I think it's nice, the observation that here he says, well, it is said that there's a considerable amount of long-range allosterine proteins, such that distant mutations coupled to the active site. Also, he mentions that there is a lack of correlation between mutations' influence on KCAT and its proximity to the active site. So if we want to, the obvious part is, okay, you want to improve KCAT, you would say, let's mutate residues in the active site and try to lower the barrier, right? But the truth is that there is a lack of correlation. So there are some mutations far away from the active site that have a high effect on KCAT. And this is in contrast to specificity. So if you want to increase specificity, then this is much related to how to active site mutations. So there's a relationship between the closer specificity and closeness to the active site. And so effective directed evolution strategies that have raised KCAT have included mutations not only in the active site, but also far away on the active site. And if you now go to the next slide, so here there's a table that shows different examples where directed evolution was applied, that the fold improvement was dramatically improve in some cases, and and also you have the notes about where the mutations are located. Most of them are distant from the active site. So this is nice because this is highlighting that these allosteric properties of the proteins, it's something that could be used for enzyme design, and that could be boosted, so can be used for selecting distal mutations. So yes, if we now move to the next slide, before getting into the is it not working? Miguel?
Yes, Silvia.
So it's not working?
It's working, but your voice is being cut too much to maybe get some sentences. So can you exit the voice channel and re-enter?
Sure. Okay, so let me just disconnect. Okay, now how is it now?
I think it's okay, at least for now.
Okay, let me know, please, if
Okay.
it's okay. I was saying that that before getting into the details of the different tools for the DINACOM, let's say, I wanted to also to highlight here this chemical review, already quite old, now quite old, but I like it.
Silvia.
Yes?
Silvia, it's not solving the problem.
It's not? So what should I do?
I have no clue, but it's difficult to understand what you're saying, so it's a problem.
Let me, okay, I'll try to go inside.
Okay.
Although, then I won't have the, okay, let me close this and I'll be back.